I'm really hyper, but very happy to have you here to talk about Eucalyptus molucana and the dieback. So I'm an assistant professor at San Diego State University, and I have done this work with my colleagues in Australia in the Hawkesbury Institute for Environment. So in 2009, Craig Allen published this paper, iconic paper, mapping all of the tree mortality around the world. You can see, you can see the points here in red, and they represent areas with high mortality. So that was 10 years ago, right? So in these 10 years, this map has become redder and redder, unfortunately, and I'm gonna make it worse today by adding this dot here of Eucalyptus molucana. Eucalyptus molucana is a dominant species in the Eastern Australia. So you can see the flowers up there being pollinated by bees, and it supports a lot of species above ground and below ground, and some of the very iconic species in Australia as well. This is the distribution of Eucalyptus molucana here in the blue dots. So it's all along the coast, the eastern coast of Australia. And we have this wide distribution encompasses 4 different barriers, geographic barriers: the Border King Gap, St. Lawrence Gap, McPherson Range, and Hunter Valley. Additionally, we have Gillison proposed to split these species in 4 subspecies. Here we have different colors, 4 different colors, and this is Crassifolia, the subspecies Crassifolia, subspecies Queenslandica, Pedicellata, and Molucani in green here. And Gillison proposed this separation based on morphological characters. For example, let's take Crassifolia, the one at the top. It has shorter capsules, a large range of inflorescence, and also a wider leaf. And the inflorescences are axillary or terminal. And what is really cool about these trees is that they peel the bark in the upper branches and the branches, and also in the trunk. They peel the bark, and he also split these subspecies based on at what point they peel the bark. So this psyllid is attacking this molucana, this tree, but it's only happening in the Sydney region around here, right? And what happens is that this psyllid has a proboscis, and the proboscis enters into the stomata of the leaves, and it sucks the sap. And then we think that this is happening in association to climate change. So with higher temperatures, these insects increase the metabolic rate, and that leads into more generations in 1 year. Instead of having 1 generation, for example, now we have 4 generations per year. But also because the winters become warmer, they don't kill the psyllids as it used to happen before. So that brings a lot of number of psyllids to the next year. Also because higher temperatures, the trees might be heat stress, right? So that also leads into this outbreak or massive outbreak. So because of this, we hypothesized that maybe the psyllid is killing the trees only in Sydney because the depleted genetic diversity occur only in Sydney due to deforestation. And I would like to remind you that the first European settlements happened recently in the 1788, right? So quite recently. And then this is a picture that represents the arrival of the British colonies into Australia. You probably cannot see, but there are eucalyptus or type of eucalyptus here, and the boats arriving. And then that happens in 1788, as I mentioned it. The first thing that the British did was clear everything, and it looked like this. Right, so all clear, arable land. Then that, with time, turned into this majestic, beautiful, gorgeous city that I love, but that doesn't have that many trees. Okay, so I have 2 goals that I'm going to present, and this is just the first goal. So the first goal, as I was elaborating, is to test the hypothesis that past land clearance following by regrowth has led to this bottleneck and reduced genetic diversity. And unfortunately, that sounded very, like I had a case for that, but I didn't find that support for that. As mentioned before, we have these 4 barriers here, and then I'm clustering all of these populations in regions, which correspond to region 1, region 2, 3, 4, 5. Sydney is here, so it belongs to the population 5. And these are different estimates of genetic diversity. You can see that, for example, if we take the heterozygosity in this region, is not a lot lower than regions 1 and 2, for example, right? So it's kind of intermediate values. But, so I wasn't sad, I need to elaborate other hypotheses, and then I came with this idea, and also because these eucalyptus are very promiscuous, that led me to elaborate that maybe the Preclearance pushed these trees that remain in the landscape to hybridize with other trees in the region. You just have to think about yourself being put in an island with a bunch of men that you could choose, but then you just have one to choose and then okay, so you got the point. So for that, I did a genomic approach. So I have close to 30,000 SNPs, and I found that I have 6 clusters here. But before that, let me explain you. I collected samples from Eucalyptus molocana and also from other species that grow in sympatry, such as Eucalyptus albens here, Bosistuana, and Crebra. So the red cluster is Eucalyptus molocana. Albens is shown in what's this color, green? Bosistuana yellow, Crebra in blue. So I have these species growing in sympatry. So, and yes, that supported the idea. For example, albens here or here, we have pure individuals of albens, and we have some individuals that share ancestry with molocana and albens as well, and other individuals, or just one, I think, of molocana, Crebra, and Bosistuana, and so on. But interestingly, it seems like we collected other individuals that have ancestry from other species that we don't know what they are. So the second goal of this talk, or this project, was to test whether the psyllid has a specificity to molocana only, or if that can be also if that could attack other species in this region. So for that, I did a common garden, and I grew individuals from populations from the different subspecies and species, and I grew them until they were like 1 meter, or 2 meters. And I put them in this landscape, which had a lot of mature trees that were being attacked by the psyllid. So hopefully you can appreciate all of these trees are defoliated and badly attacked. Some of them were about to die. So this is the psyllid. Here I was looking at the presence of females. They're the big ones, the males, and then also how they lay the eggs, and I follow them during the development. You can see this is how they look in the field. I was looking at the males. You can identify them by, they are smaller and they have this appendage at the end. And then we have the very big females, and then we were waiting for them to mate, and sometimes that happened, but other times it didn't happen because there were some intruding guys coming along. So as I mentioned, I collected populations from the 4 varieties or subspecies that I introduced to you. I also included species that were closely related to Eucalyptus molocana. So Eucalyptus molocana, Microcarpa, albens, and Wolfsiana belong to this supraspecies molocanae, superspecies. So these are closely related. And also I included other species distantly related, but still belonging to the series Buxales. And these are in my map. Now they are showing in blue. So these are the provenances that I took. And as I mentioned, this is how they look in the field. And these are the results. So you can see when I only look at the visitation of females and males, you can see that they tend to visit mainly Moluccana in both cases, Eucalyptus moluccana, but they also visit the other species. But then when we look at the eggs, the oviposition, really what's going to happen to these trees, we saw that Moluccana again had a lot of eggs, and few individuals of Wolfsiana and albens had also presence of eggs. So just to remind you, these are species that belong to the superspecies Moluccana. And I didn't present the results. I'm not going to present you the results here, but just to summarize, I have proof that these different species are able to hybridize. So now I'm really interested in seeing what genes might be moving around, and maybe these are potential hybrids that they have genes that confer predisposition to the psyllids. But very interestingly, when I look at the subspecies level, this is only Moluccana, the 4 subspecies that I introduced, you can see that Eucalyptus moluccana variety or subspecies Moluccana, the one that is being heavily attacked, actually had lower percentage of eggs compared to the northern populations. And this is really worrying because that tells me that if this psyllid, which I don't remember now if I mentioned that it was native to the forest, but it's native to the forest, but right now only present in the southern populations. If it goes to the north, it might cause a big troubles to this forest. I think this is my last slide. So just to summarize, I have mentioned that Sydney has high levels of genetic diversity, but we need to look under the lens of hybridization to explain that. Also that the susceptibility was very high in Moluccana, but there were also potential hybrids that also were susceptible to the psyllid, and the varieties in the north are more susceptible, which again might be a challenge for for those populations. I would like to acknowledge a lot of people that have helped me through these years to work in this project, and if I can take any questions, just in case that you wanted to see the meeting.